Good afternoon. I'm sorry that I'm not able to be with you this afternoon. Today what I want to focus on is how we're going to be building soil by managing carbon flows. The carbon is truly the key to how we're going to get these systems functioning and be able to maintain resilience and activity that we're looking for. And the way that we're going to be doing this is really regenerating soils. Soil itself is carbon, hydrogen, and oxygen, or organic matter, bound to sand, silt, and clay. But we're looking at how we're going to regenerate soils. We can look to regenerative agriculture and the regenerative agricultural approach. This is a systems approach where everything is integrated, working together on both the macroscopic and the microscopic scales. I define regenerative agriculture as a dynamic, innovative, integrated, intensive system that is going to be focusing on photosynthetic activity and carbon flow, the carbon economy, where things are paid for based on utilizing a carbon currency. Functions that we're looking for from the soil are actually purchased from the soil biological community with this carbon currency. So what we really want to do is we don't want to have a new green revolution. We don't want to have a plant-based revolution as much as a brown revolution, a soil revolution that's looking at ecofunctional intensification across your entire farmscape. It isn't honing in on one particular activity or one particular thing that you could do, one particular system, raising crops or raising animals, but rather integrating again across the entire landscape, across your entire farmscape, where we're going to be optimizing landscape use. We're going to be thinking about how we can maximize the efficiencies, in particular for water and nutrient use efficiencies. We're not going to be looking at driving the system in an intensive manner to do more, but to do less by working with these efficiencies and driving the system. Intensification doesn't mean that it has to be more. It means that we need to strategically determine how it is that we could achieve more with less. This may include things like multiple enterprises, but the more important thing that we're looking at is because we're looking at spreading the risk among all of the microscopic and macroscopic organisms that are a part of this system, that's going to reduce the individual risk to one single individual, including the farmer or rancher. It's taking that risk and leveraging the functionality that's happening on the macroscopic and microscopic scales. And that's going to help us to improve the nutrient density of what it is that we're growing. And we're looking at this from, again, a sort of a cost-benefit ratio, where everything costs, but if we do if we distribute the cost among a number of different organisms and are doing that to maximize the efficiencies, that's going to be able to leverage the opportunities that we're looking for from sunlight, air, plants, water, soil, birds, and microorganisms. So we really just need to think like Snoopy. For us to be doing this, one of the things that we're going to be looking at and thinking like Snoopy is creating a new understanding of soil organic matter. I'm a soil microbiologist. I have a doctorate in soil science, and I was taught in soil science classes that it takes 1,000 years to generate an inch of topsoil, and that you can't change organic matter percentages in your lifetime. And that was the traditional viewpoint that we had on organic matter, that organic matter was decomposed, humified, plant and animal debris. But what we're finding in this emergent view, scientists are beginning to understand the roles that the microbial community and biology play in changing the way in which organic matter is stabilized. So we're looking at organic matter that is going through back and forth through a continuum of different types of transformations. This is referred to as the soil carbon continuum model. And that's what it is that we're looking for, is how soil and organic matter is flowing through this whole entire system. So when we do that, we can take a look at sort of this traditional view of soil organic matter composition. Soil organic matter itself is a very small amount of the mineral component of soil, but it controls about 90 to 95% of the functions that are happening in the soil. And you have it primarily, it's thought of being composed of a small amount of microbial biomass that contains fungi and bacteria, microarthropods or microscopic animals. You have protozoa, nematodes, algae, all of these different organisms that are a part of this. And then you have the labile or particulate organic matter that is coming in on an annual basis, primarily plant debris or animal debris. And then you have this stabilized fraction, that humus fraction, that is said to be so humidified by the process of decomposition. But what we're really seeing in this regenerative approach is that we can generate topsoil in less than 1,000 years, and we can change organic matter percentages in less than a lifetime. And we're doing that not because we're recreating soil and not because we're recreating organic matter from the initial mineral components and the biological activity, first land plants. That isn't what it is that we're looking at. We're actually looking at a process of regeneration, which is going to be something that can happen in a shorter period of time. And within this process of regeneration, one of the biggest things that we're looking at here is how the microbial community is transforming that labile fraction and making it stabilized in biological, chemical, physical, or geological stabilization mechanisms. So binding to minerals like clay minerals, being occluded inside soil aggregates, changing the chemical nature through decomposition processes or through the creation of various types of complexes in order to make it less available to enzymatic reaction, that organic matter. And that's what's going to be able to help to build up the real organic matter over time, and where we can start to see the changes in the percentage of organic matter that we're measuring in our soils. So it's important for us to think of this again as a regeneration model, not as a creation model, not as something that happened with the first land plants in a mineral environment before we even had soil. When we're taking this approach, what we can find is that various types of tools and practices can help with this regenerative process. So what you have here are two soils. One soil is from David Brandt's farm near Carroll, Ohio, and the other soil is across the road, or across the fence line, I should say, from David Brandt's farm. So the soil that's light brown that's at the top is actually that soil from the other side of the fence row. And the soil that's dark and rich is what David Brandt has been able to regenerate by getting that influx of carbon, organic matter, going into that mineral environment and building up soil, regenerating soil again. So you've got that dark richness that David Brandt has created over 30 years. Now what we can look at are some of the tools and practices and ideas and concepts that David Brandt utilized, and actually figure out how we can optimize the efficiencies to be able to get this regeneration of soil in a shorter period of time on our operations. And we need to do this because as you look at these pictures are dust storms. And the question is when were these pictures taken? Were they taken in the 1900s or were they taken in the 2000s? What do you think? Which one was in the which ones or one was in the 1900s and which one or ones was in the 2000s? In reality, the only one that was in the 1900s, and it was around the 1930s, is the picture that is in the lower right-hand side. picture of the road where you can see the dust cloud that's there. The other pictures were actually taken since 2010. We have one picture that is in the lower left that was taken in 2012, and you have a picture, the pictures that are in the upper right, that picture was taken in 2020, and the other picture was taken in 2021, just last December. So very recently. And when we look at these pictures again, we've told ourselves that the Dust Bowl was never going to happen again. We were never going to be getting this loss of soil from our agricultural fields. We created the Soil Conservation Service, which became the Natural Resources Conservation Service. We've implemented various types of conservation practices and policies so that we wouldn't have the Dust Bowl era again. But when you look at these pictures, we still have it. It's still happening in this system. And so what we need to do is figure out what tools we can put into place to help to build the resilience of the system and be able to keep that soil in place on our landscape. And that's what we're trying to do with regenerative agriculture. So this is sort of some other pictures that I took just recently. My family, my dad farms near Lake Benton, Minnesota, just inside the South Dakota border or just inside the Minnesota border, I should say, from South Dakota, from Brookings, South Dakota on Highway 14. And if you're going from Highway 14 onto Highway 12 to head up to Hendricks, Minnesota, I pulled over the other day when I was driving up to Hendricks, and I took these pictures, one from the west side and one from the east side. And as you can see on the west side, where they tilled this field in the fall, there's a lot of snert that has blown into the ditches that you can see there. But on the east side, it wasn't that the wind got stopped in any way, shape, or form. It's just that the wind was not going over that fine dust that was created by tillage, but instead the soil is protected by the armor of the plants that are growing in that area. So you don't have that same level of soil loss in that environment. And a friend of mine, Steve Groff, many of you probably know of Steve. He has done a lot of work with cover cropping in Pennsylvania. And Steve Groff, a few years back, decided to collect on January 7th some snert, this combination of snow and dirt that blew into the ditches on his neighbor's field. He went out there and he collected some snert and he sent it in for nutrient analysis. And as you can see, it's actually very high in phosphorus and potassium and has optimum or high levels of pretty much every other nutrient. If our issue is that we don't have enough fertility to meet our yield goals, why is it that we're actually allowing for our nutrients to blow into our ditches and create snert? We need to be thinking about what it is that we can do to preserve not just the soil, but the quality of that soil, that nutrient density that's in the soil that we then want to achieve in the plants that we're growing and the animals that it is that we're raising on our landscape. So when we look at what's happening to the soil environment, we're still in an environment in which we've got very high global erosion rates. So the colors that are dark green to yellow, yellow is 0, and then that is basically the loss at dark green. It's a loss of more than 3 tons per hectare per year of soil loss. And if it is in a reddish color, that is actually not a loss. You're actually keeping 3 tons per hectare per year of soil in that environment. So what we've got here is we've got this whole spectrum of erosion that we've got happening in these soils. And you can see that there is a very high loss of soil that's happening in some areas of South America and in a couple places in Africa and Asia. But the United States also has a fairly high loss of topsoil every year on every acre, on every hectare. In fact, in the US, our erosion rate of sheet and rill erosion or water erosion and water and wind erosion is about 4.63 tons per acre per year. That's what we're losing every year on every acre in the United States. Now in 1992, that was up at 7.13. It's come down to around 5. But since 1997, for the last 20 plus years, this value has not really changed. Between 1992 and 1997, we started increasing some of our conservation practices. We saw more use of things like no-till or conservation tillage. We saw strip tillage, these different types of practices starting to come into use. But then as we got into the 2000s, that change, that shift that happened with tillage and that level of conservation has been kind of static in how it's been able to protect our soils. So there's something more that we need to do than just be focusing on tillage. This is not something that we can continue to afford to be doing, this level of soil loss on our landscape. If you were to multiply this by the number of acres that are in agricultural production in the US, basically we lose about 1.67 billion, with a B, metric tons of topsoil every year in the United States. If you were to load that soil onto rail cars, onto into cars, rail cars, 100 tons in each car, the length of the train would actually circle the Earth more than 8 times. Again, we lose nearly 1.7 billion metric tons, billion with a B, metric tons of topsoil in the United States every year. This is not an acceptable soil loss. We have conservation programs that talk about acceptable soil loss. How can soil loss be acceptable? We're supposed to be in the process of regenerating the soil, not losing the soil. In addition, as we saw with the snert, you're going to be losing fertility. When we lose soil, we're also losing some of that structure that can help us with holding on to water. So we're going to start to see an increased need to do irrigation. And as we increase the amount of irrigation that we're doing over time, one of the other issues we're going to also start to be seeing even more is issues with salinity. This is in Saskatchewan this last year in 2021. And what we're seeing here is that we're not managing the hydrology of the prairie pothole region very well, meaning that we aren't allowing for the water to go down deeper into the soil profile and be held there with the roots drawing that water down. Instead, the water is sitting more on the surface and evaporating off, leaving behind the salts. We need to be looking at how we can manage water much more efficiently at the same time that we're looking at how we're going to be managing nutrients. So what we have here is a global picture of the fertilizer use per hectare of cropland in 2018. This is looking at the big ones, NPK. And what we've got here is this idea that in the United States, we're somewhere around roughly about 250 to 300 kilograms of fertilizer that we're using on a per hectare basis of cropland. That rivals some of the other areas in the world like China and some places in Europe. But we're looking at something in which it's a use that we can no longer continue to afford to do. When you look at just that fertilizer use globally, you can see that we've been increasing our nitrogen use, our phosphorus use, and our potassium use since around 1960. We've seen this huge bump in fertilizer use, that addition of fertilizer. Now, there's two things I want to point out here. One is this is started around 1960, which doesn't mean that we haven't always been adding fertilizer to our cropland. When we have that notion that it's always been done, so we have to continue to do it, we need to fight against that notion, and we need to look at what the real data shows as far as when we started really using some of these synthetic fertilizers that we've been adding to our soil. Because this really goes to how the system is going to be functioning for nutrient use efficiencies. When we think about pesticide use, we're also a country that has a fairly high pesticide use per hectare of cropland. Again, going down and looking specifically at the breakdown of various types of pesticides in the United States, most of it is herbicides. We use quite a few insecticides, some fungicides and bactericides, and some other pesticides as well. So the whole idea here is that we're taking away some of the functions of the soil by replacing that with pesticides and fertilizers that we're adding. We're not looking at the system as a whole, but we're doing replacement technology. And that's not going to be a thing that's going to allow us to regenerate soil, because soil regeneration is based on carbon flows, that carbon getting into that environment, and that is going to be triggered by enhancing the functionality of the soil. How is this all working, and why is this all really, really important? Well, for me, these are the people that make it very important in my life. The first picture that you will see is of my younger sister. She's disabled, has been disabled for the last 40 years. But you can see her looking at our new grandniece, and that grandniece, those two staring sweetly at each other. Those are the people that I'm most concerned about. In addition, in the middle picture is one of my nieces and the very youngest new grandniece. So the niece, my grandniece in the first picture, she is now going to be 5 years old this year, but you can see in this other picture that she is got a younger sister now. Her younger sister was who I was visiting as I've been here in Minnesota. That younger sister is about 7 days old, so about a week old now. And then in the other picture is that grandniece's older brother and his dad. And these are the people that I am really concerned about, and why I continue to look at how we could be able to set up a system to be farming forever. And being able to regenerate the soils is a key to that, and that carbon flow. The other reason why I wanted to point out these individuals is where they're living in Minnesota and the southeastern corner of South Dakota. In that area, the groundwater that supplies the water supply for about 65,000 people has had to change recently because the well is contaminated with agricultural nitrates. And so they've had to search out to find a new wellhead to provide water for around 65,000 people. And that is something that, again, we need to be looking at how we can improve nutrient use efficiencies and how we can regenerate soil, because the carbon that is a part of soil is one of the best filters on the planet for being able to produce clean air and clean water. That's how we have clean air and clean water. But if in a commercial level, if you have a Brita filter or a pure filtration system or an air filter, one of the filters that they will have in those filtration systems, the primary filter, is actually a carbon-based filter, because carbon is really good at binding things and holding on to them. And that's what is happening in the soil, in a way that that carbon and organic matter is holding on to the different types of nutrients that are in the soil and filtering that out before that water gets down deeper into the soil as part of our groundwater resources. I've been able to travel all throughout the world, and one of the things that I am completely awed by is the fact that all of us share this united, universal soil perspective, that soil foundation to agriculture. It is the engine room of productivity. It is the way in which carbon flows are going to be happening. Organic matter, that carbon moving into the soil and it being transformed by the biological community and providing for the various functions that we're looking for from our soil environment. So what is it that we could do within this brown revolution, within this ecofunctional intensification? What are some of the practices that we could put into place, or the tools that we could put into place? I'm not going to discuss specific tools. I know that you've probably heard about various types of tools at this conference, but what I want to do is discuss this from the framework of a pyramid that is going to be providing the principles to economic and environmental wealth. So what you have here are 6 soil health principles. Some people will talk about 4 soil health principles or 5 soil health principles. What I've got here is 6. And the reason that we've got 6 is because this is really principles that are focused on soil carbon flow and how to be able to manage that flow. And these principles, I've put them in this pyramid design. And in this pyramid design, I did this because every block that was laid in a pyramid was important. It played a role and interacted with the blocks that were all around it. And those blocks I think of as the various tools and practices that we can put into place to address these principles. But we want to be looking at how they're integrated with each other in order to allow the pyramid to stand for thousands of years, or in this case, for your farmscape to stand for thousands of years. Instead of celebrating century anniversaries, I want us to be celebrating millennial anniversaries on our farms and ranches. And we can do this by thinking about how we're going to be managing carbon flows. So looking at getting that carbon going below ground, maximizing photosynthetic activity, essentially maximizing photosynthetic time. Part of this is not really looking at increasing your seeding rate or increasing the amount of photosynthesis you have at one point in time, but it's actually making sure that we're putting carbon below ground as many days of the year as you possibly can. We don't want the soil and the soil biology to have to go through periods of feast or famine. We want to make sure that we've got that carbon flowing below ground as many days as we possibly can. So here in the Dakotas and in the upper Midwest, we need to be thinking about the fact that our job, first off, is to harvest sunlight. We're going to put tools and practices into place to help to get that sunlight harvest happening at the most efficient rates. We're going to have those tools and practices in place. And what we're going to be doing with that is really looking at how the carbon is going to be flowing through this system by making sure that we've got carbon flowing as many days of the year. And in this area, what that means by as many days of the year is that there's at least 260, if not 280 days in which you need to be putting carbon below ground. This doesn't mean that every plant that you have growing is going to be growing to harvest and to yield. but it is going to be growing to get that carbon flowing. So rather than actually having this happen where you've got a plant that's going to be growing until you can harvest it, you're actually harvesting sunlight. That's the key that you're looking at, not harvesting grain or harvesting forage, but harvesting sunlight and getting that carbon flowing below ground. In order to be able to achieve this, you're going to have diversity in the plants that you have growing. That diversity is going to take advantage of the plant's capabilities to be growing at different times of the year, to be more frost tolerant, to grow at times when it's really cold or at times when it's really hot. Plants do not have a nervous system. They're not like us. They don't sense temperature in the same way that we do. What temperature does is temperature controls the reaction rates of their biochemistry. And plants that have adapted to different temperatures have different reaction rates that are optimum at those various temperatures. So what we want to be looking at is really what are the plants that we can put into place to optimize these efficiencies. That diversity is going to be in the form of the plants, but it also is going to help in the form of other types of macroscopic organisms. It's going to trigger diversity in wildlife, in birds and bats and bees. It's going to trigger diversity in insect populations. It's going to trigger diversity in microbial populations. So we're going to have both microscopic and macroscopic diversity. We want to be able to look at also how we're going to manage the function of those biological organisms. We're going to get carbon to them. We're going to have them be in diverse compositions and consortia to work together. But then we want to make sure that they actually have a function or a job to do. And that function is actually managed by how we're going to be managing the inputs that we put into the system, in particular the synthetic inputs like fertilizers and pesticides. What happens when we add those chemicals to the soil environment is they take the jobs of the microbial community. And when the microbial community does not have a job anymore, most of the microbes are involved either directly or indirectly in getting nutrients for the plant. And when the plant has all the nutrients it needs, those microbes don't have a job anymore. And what ends up happening is that those jobs get outsourced and the microbial community doesn't get fed. There isn't carbon going below ground to that microbial community. The plant says, I don't need to trade you. I don't need to pay for nutrients from you if I already have those nutrients. So what I'm going to do here is I'm actually just going to keep the carbon to myself. I'm not going to give off those exudates in order to be working with the biological community. And when those jobs are outsourced, again, the microbial community doesn't get fed, it doesn't reproduce, and it doesn't continue on with the function even after those synthetic nutrients are gone. They're either immobilized or they've been leached out, they've run off, or they've been volatilized into gases. So you don't have that fertility that you added at one point in time maintaining itself throughout the whole growing season. The same is true for various types of pesticides. They have a certain window in which they work, and then afterwards they're not working at the level that they could be. But the microbial community has multiple mechanisms to be able to manage pest and disease issues. It isn't just one pathway that the chemistry follows. This is why we have to keep changing the chemistry, because there's only one pathway that it uses to actually manage pest and disease relationships. The biological community uses multiple pathways in order to be able to do that. So if we can reduce those inputs, we can keep the microbial community functioning. The next thing we want to do is manage livestock. And this livestock is not necessarily just a large grazing animal, but this livestock is animals at all levels, microscopic to macroscopic. managing the landscape in some areas to attract predatory insects, managing the landscape in another area or in the same area to attract pollinators, to attract bats and birds, to be managing wildlife in such a way that we can actually, with the livestock, what we're trying to do is we are trying to drive a certain amount of reaction on the part of the plant. The plant responds to that pressure that's coming from the livestock, from those animals at all levels, from microscopic to macroscopic, that do a certain amount of damage to the plant tissue. That damage is a stress event. The plant then responds by forming various types of stress biomolecules, things like antioxidants and polyphenolics and other phytochemicals. To be able to make those phytochemicals, as well as the enzymes that are required to do photosynthesis so that the plant can continue to grow and regrow from that injury, the plant needs various types of micronutrients from the soil environment. That's going to trigger the plant to give off exudates to get the microbial community to function, to release those nutrients, put them in forms that the plant can absorb or that could be absorbed by the mycorrhizal fungi and transported to the plant. So when we're looking at this, again, it's about managing the whole system. We also want to manage the system to reduce soil disturbance and to reduce tillage. As we could see from some of the erosion numbers, we were able to reduce some of our soil loss by managing tillage. But one of the things that happens is that just managing tillage is not enough. We actually need to have another layer of influx that's bringing carbon into the soil environment. Tillage, or the lack thereof, is about managing loss. What we want to do is we need to regenerate and re-engineer that soil environment to keep the soil in place, not just to manage the loss, but to actually keep it intact and in place. And then last principle that we have here is that armor or the protection for the soil surface. Making sure that in that picture of the snert that we had from Hendricks, where we had the soil that was blown off from that till field that was bare, that allowed that soil to get blown off into the ditch and form snert, that combination of snow and dirt. Whereas, and the other side of the road, on the east side, the soil was protected. It had an armor protection there. It had standing plants that were there. It had the plant tissue that was on the surface that was creating a duff layer. All of that is going to be a part of that protection. You have things like cover crops that can also be part of that protection and that armor. Something that's going to diffuse the energy of wind or water from hitting that soil surface, and even the energy of solar radiation from hitting that soil surface and breaking down the organic matter. When we're thinking about the various tools that we can put into place, the way that we're going to be managing nutrients and nitrogen and grazing and tillage, adding diversity, how we're going to manage pesticides, one of the things that we should be thinking about is this FIST acronym, the frequency, intensity, scale, and timing. The tool or the practice that I'm using, how is that going to impact the frequency of how many times am I going to have to continue to use that? And is that a positive or a negative? If we split applications of nutrients, which means that we're adding them more frequently, that can be a positive. But if we do a number of tillage passes, doing tillage more frequently, that is a negative. So thinking about what is going on with the frequency, what is the impact of the frequency of this practice? What is the impact of the intensity and the scale? How much of the total soil volume am I impacting? How tough or hard or fast am I impacting that? When it comes to nutrients, What is the scale or the quantity of the nutrients that I'm adding at one point in time? And is that good or bad? With tillage, what is the tillage tool I'm using? How much of the total soil volume am I impacting? If I impact more of the total soil volume, but it reduces my frequency, maybe there's an overall benefit for that. And in fact, there probably is, because the system can recover from one tillage pass or a few tillage passes very infrequently. But if that is that repeated event, that is when the system starts to have an issue. It doesn't have that recovery time. Again, thinking about this from a grazing aspect, when we're talking about recovery time, that frequency, that intensity, that scale. Also looking at timing. When is this tool going to have the least amount of impact? Negative impact. In some cases, like with grazing and managing weeds or being able to manage some surface compaction, you can use grazing as a tool. Hoof traffic and grazing and consuming of those weed pests that are where you're timing it in order to be able to do that. In some cases, you don't want to time it where it's going to have that level of soil disturbance. So thinking about the tools and the practices that we put into place from the aspect of frequency, intensity, scale, and timing when it is that we're choosing what tools to use. Now, when we utilize regenerative agricultural practices, one of the things that we find is that we're building up resilience in the system. We're making the system function at a much higher rate with much greater levels of efficiency. And what this does is this can help to improve our water use and our nutrient use. And when we're talking about improving water use, it is improving water infiltration rates and water holding capacity, that ability to store water in the rhizosphere and the ability to get water in when it rains very heavily in a short period of time. So this is an example, many of you have probably seen this picture before, of Gabe Brown's ranch in 2009 when we had a precipitation event where we had about 13.6 inches of rainfall in about a 24-hour period. And at this precipitation event, this picture was taken the next day. What Gabe has done is he's been able over 20 years or less to change his infiltration rate from a half an inch an hour to about 8 inches an hour. That allowed for him to be able to absorb this 13 inches of precipitation over a 24-hour period. When we're talking about improving infiltration rates, this is connected to soil porosity, the open space between the solids in the soil. And that open space should be about 50% of the soil volume. It should be about 50% solid, 50% open space, with half of that open space filled with water and half of that open space filled with air. So what happens is if we can increase our porosity, if we can get that space more in a certain area, that can increase our infiltration rate of the first inch of water by 167% and of the second inch of water by 650%. And that's really critical to what it is that we're looking for from our system, that building of resilience, taking in the water and being able to hold on to the water. So again, this illustrates this whole idea of porosity, that open space that's there. And you want that open space to be filled with both water and air, because the plants, just like us, are aerobic. They take in oxygen and give off CO2. Now I know what you're thinking, wait. I thought that plants took in CO2 and gave off oxygen. And they do that via photosynthesis. They do that in their foliar tissue above ground. But they also do biochemical reactions in those cells and in their leaves and in their roots that are aerobic biological biochemical reactions that require oxygen to drive those reactions and as a result, produce CO2. In the rooting environment, if you don't have adequate porosity, where you don't have enough air coming in, so in that unhealthy soil that you see there, you have more solids than what you had in the healthy soil, but you have about the same, if not a little bit more water, but what you're really missing is airspace. That air, again, if you don't have that ability to have a good gas exchange, what happens is that as the roots give off CO2, that CO2 can't escape and starts to build up around the roots and results in an anaerobic environment that starts to suffocate the plants. This is what we'll see happening in very saturated soils, is that the plants can't get oxygen that they need, and their CO2 is building up in that soil environment. Because the gases can't diffuse well through the water to be able to get through the roots, the oxygen can't get to the roots, and the CO2 can't escape. When we're looking at porosity, again, what we have is the ability of the biological communities to actually influence porosity by helping to form soil aggregates, these pellets that are in the soil, these balls that are in the soil that give the soil this cottage cheese type of texture that you have in there. And so what you see here are 1 to 2 millimeter aggregates. They were separated by passing the soil through a series of screens, with the top screen having an opening of 2 millimeters and the bottom screen having an opening of 1 millimeter, and collecting what passes through the 2 millimeter screen and what gets collected on the 1 millimeter screen. Now, the biology can help with the formation of these aggregates and the stabilization of these aggregates. And what's important about the aggregates in creating pore space is if you put these aggregates of different shapes and sizes together, they don't fit tightly against each other. There's actually open space between them, as you can see in that middle photograph, where you've got smaller aggregates and medium aggregates and large size aggregates. And what that allows, the arrows illustrate, is that water or oxygen can flow around those aggregates and into that pore space. The soil core profile that you see in the far right picture, that's indicating this is a very fine sandy soil up in Alberta. And what you have here is you can see that pore space that was created by the biological activity in the first about 1.5 feet of that soil core. But then as you get below that, you're now starting to see that fine sand filling in that pore space. And when that fills in that pore space and you don't have as much pore space and opening, again, you're not going to get the good gas exchange, and you're not going to have a lot of water infiltration. The other thing that happens with the aggregates, and these arrows really illustrate this, is that the path that the water takes is not a straight path. It actually bends and curves and moves around the aggregates. And what this does is that keeps more of the water between those aggregates in the rhizosphere zone. This would be like setting up a plumbing system in your house that, instead of going from a 6-inch pipe that you would have coming into the house and then maybe going to a 2-inch pipe and then a 1-inch and a 1/2 inch, essentially what you're doing is you're going from a 6-inch to a 1/2 inch to a 2-inch to a 1/2 inch to a 1-inch to a U-shaped 1-inch to another 6-inch, and all of these things, with all of the changes in the size, the diameter between the aggregates in that path, is going to keep the water there for a longer period of time. So again, thinking of this plumbing system in your house, if you did that with the pipes, what would happen is you wouldn't get good flow, because it would take too much pressure to try and push that water through that piping system with all of those changes in diameter. What we're trying to do is we're trying to keep the water between those aggregates in the rhizosphere, instead of gravity pulling the water down and or evaporation, solar energy, pulling the water off of the surface. When we do this with biology, we can also do this on a much larger scale than just with the aggregates. You have earthworm tunnels, and this is a picture of a Blake Vince, farmer from Ontario, and these are the earthworm tunnels that he has in his field. Blake has been working on creating his system so that he's an earthworm feeder. Again, a sunlight harvester that is going to be putting that carbon below ground, giving that to those earthworms so that they create these complex tunnels that are creating, that's that pore space that he has there. That's all the porosity that he's creating around those earthworm tunnels in his soil. Think about the amount of water that you could absorb and hold on to in that environment. This is what soil aggregates are going to be looking like, that are going to be this other contribution to pore structure. So we've got things like earthworm tunnels that can contribute to pore structure, and then we've got these soil aggregates. And what you see here are aggregates of different shapes and sizes, and they can be formed in a relatively short period of time. So the two pictures on the bottom are aggregates that have been formed on roots. The picture on the right-hand side is aggregates that were formed in about 3 months in a pot culture experiment. And the aggregates on the left-hand side were formed in about 60 days on a millet root that was planted as a cover crop mixture following a forage pea harvest. And this was done in North Dakota, in a field in North Dakota. So you can see that you can actually get this aggregate formation in a relatively short period of time. But just because you have aggregates formed doesn't mean that you can keep the aggregates intact. So between the space of the particles that are a part of aggregates, you have silt particles, you have sand particles, you have clay minerals, you have pieces of organic matter, you have bacterial colonies, you have fungal hyphae, you have all of this stuff inside a soil aggregate. That's all a part of creating that aggregate. And in between all of those particles is open space. When the aggregate is dry, that open space is going to be filled with air, with gas. When it rains and water starts to flow into the soil by capillary action, that's going to rush into the aggregate. And again, because of density, the water can move more quickly than the air can escape. So what ends up happening is the aggregates get pushed, the air molecules get pushed closer and closer together, and the aggregate literally eventually explodes. And so you lose all of the carbon energy that went into creating that aggregate. You get that explosive force that's happening. And when you do that, those fine particles that were once inside the aggregate are now going to be more susceptible to erosive loss through water erosion or loss through wind erosion. What we want to do is we want to get the biology to take carbon that's flowing below ground and not just form the aggregate, but also to help to stabilize the aggregate. So what the biology does is the biology will put sort of a weather sealant on the outside of the aggregate. This is similar to putting a weather sealant on the outside of your house. That weather sealant can allow for gases to move, oxygen to move in, and CO2 to escape. If this wasn't true in our houses, we all would basically suffocate because we wouldn't be able to get that gas exchange. So our houses, that weather sealant and that structure is porous enough to allow for the gas exchange, but not to allow water to enter. So what happens is the water is going to be moving in at a much slower rate, which gives time for the air molecules to escape and or dissolve in the water. And now that pore space that was inside the aggregate is going to be water filled instead of air filled. And it's going to be stabilized and stick around for a longer period of time. And we can see this in the real world. So these were aggregates that were separated from soil in an experiment that was conducted up in North Dakota. And what you have here is those aggregates that were collected by passing the soil through a series of screens. These are the 1 to 2 millimeter aggregates. So they pass through a screen with a 2 millimeter opening and were collected on a screen with a 1 millimeter opening. And we put them in glass dishes, and then all that was done was the lower pictures are just after we added water. And that's that capillary action of the water coming in and breaking up those aggregates. So in the first column of pictures, that is from a conventionally tilled spring wheat fallow system, and the WSA is a percentage of the amount of those aggregates that stay together. So very few of those aggregates, as you can see, stay together. The middle is from a no-till spring wheat, winter wheat sunflower system, and roughly about half of those aggregates stay together. And these two plots were set up in an experimental trial where they were next to each other. So they were under the same climatic conditions, they had the same parent material, they had the same amount of time for the soils to be created and have evolved. They have the same topography, but what they don't have is the same amount of biological activity that's being triggered by the organisms that are there. Again, they have the same topography, they've been around for the same amount of time, they have the same type of climate, and they have the same type of parent material. So everything in the soil forming factors, the five soil forming factors was the same, except the biology and how the soils were managed, the biological activity. Now, the difference between the middle one and the last one is this moderately grazed pasture that was separated from those two plots by maybe about a half mile. So again, those four soil forming factors were the same, but what you have in this last one was you had plants that were growing almost every day of the year. As many days as you possibly could, you didn't have this feast and famine reaction that was putting the biology to work and providing carbon to that biology on just a few days of the year. Instead, that biology was getting fed more days of the year, which provided for the carbon that was used to make the weather sealant on the surface of the aggregate. So what ends up happening in the process of aggregate formation, the spectrum of aggregate formation, is that you have carbon that goes to the microorganisms. It stimulates their growth. They start to help to form soil aggregates. They'll make polysaccharides that will help to glue those aggregates together, but they need another sort of flush of carbon in order to be able to make that weather sealant. So if every year they only get enough carbon to reproduce and grow, they're not going to really make very good aggregates. If they get enough carbon to reproduce and grow and make the polysaccharides and start to glue the aggregates together, that's good, but now they're still limited by the fact that in that process they're not getting enough carbon to make the water stabilizing molecules. So when we're looking at how it is that we can manage carbon flows, what we really want to do is make sure that as much as possible, carbon is flowing below ground and feeding the biology, that we're not outsourcing the jobs to the microbial community, and we're not losing that functionality of the soil to form the soil structure, the aggregates that are going to help with porosity, that's going to help with aeration, and with water use efficiencies. And in addition, inside those aggregates, the microbial community, the bacteria and fungi that are existing inside those aggregates now have ready access to the nutrients that are released from the minerals that are inside the aggregate, as well as the organic matter that is slowly broken down by that bacteria. So it's a way of improving nutrient use efficiencies because the aggregate acts like a mini grocery store where the microbial community, the bacteria and fungi that are inside that aggregate can help to stock the shelves of the grocery store, and the mycorrhizal fungi that are growing from the inside of the aggregate out to the plant roots can transport the nutrients off of those shelves and deliver those nutrients right inside the root cell wall up to the front door of the cell membrane. Very efficient delivery of nutrients. Again, keeping everything in that aggregate grocery store. So all of these become layers in which the microbial community is utilizing the carbon and carbon flows that are coming from the plant in order to be able to address the various types of issues that the plant may have so that it can improve the resilience of the overall system. Well-structured soil, better water and nutrient use efficiencies, better pest management, all of these things are dependent upon that carbon flow. So with that, I want to thank you for your time and attention.